We're continuing our studies in Chapter 22 on protein synthesis, and in this lesson we'll be briefly reviewing how we translate the message. Remember, our translator is tRNA. Now the T stands for transfer, but you can think of that as translator. The anticodon in the tRNA is going to base pair with the codon in the message, and that's part of our illustration here. In this case, the tRNA is pictured in its two-dimensional form so you can more readily see the interaction between the anticodon and codon. However, always keep in mind the actual form in the cell is the three-dimensional shape, that L-shaped structure in our smaller box here. Of course, the amino acid is attached at the 3' end. This will be the amino acid that will be delivered. So in this case, we have the message at the bottom of the screen here. It's going to be translated in the 5' to 3' direction. That was the direction in which it was synthesized. In this case, we're looking at the universal start codon AUG that specifies the amino acid methionine. The tRNA in its anticodon needs to base pair with this codon, and so the anticodon sequence will be UAC. Keep in mind, however, since the message is in the 5' to 3' direction, the anticodon needs to be the reverse complement. In other words, in the 3' to 5' direction, UAC. You can see this more readily if you look at the tRNA molecule. Here's the 5' end on the right. On the left is the 3' end. So again, if we read that anticodon sequence UAC, we're reading it in the 3' to 5' direction so that it can base pair with the codon in the 5' to 3' direction. Recall that we have 61 codons to specify amino acids. Do we need, therefore, 61 different tRNA molecules? No, remember we have those isoacceptor tRNAs, and they can bind multiple codons. We'll see how that works in just a moment. Remember, an isoacceptor tRNA is a tRNA that has different anticodon, but it accepts or specifies the same amino acid. This is all due to that wobble hypothesis, where the third base in the codon wobbles in terms of the difference in nucleotide, and yet it specifies the same amino acid. This is the degenerate nature of our genetic code. And so one tRNA molecule can bind multiple codons, and that means we need fewer tRNAs than there are codons. Let's see how that works. Let's look at the example of the anticodon of the tRNA molecule that specifies alanine. Its anticodon in the 3' to 5' direction is CGI. I is the specialized nucleotide inosine. And for your convenience, I've included an illustration of the structure here. Let's compare that to adenosine. As you can see, inosine simply results from the deamination of adenosine. That's the only difference between them. And because of the difference in the chemical structure of the inosine nucleotide, it can base pair with either U, C, or A. So you can see how a tRNA carrying this anticodon CGI can base pair with multiple codons and yet deliver the same amino acid. And that's illustrated at the bottom of the screen here, the base pairing interactions between the anticodon of the tRNA and the codon of the mRNA. Always keep in mind the third base in the codon is base pairing with the first base in the anticodon. So the wobble hypothesis explains why many codons can be bound with fewer tRNAs. Here's a table from your book illustrating the allowed wobble pairs at that third codon that base pairs with the first base in the anticodon. So we have the anticodon base in the 5' end on the left column and the 3' base in the codon on the right. It's not important you remember the details of this table, simply that the third base wobble allows for this multiple codon recognition, and that means we need fewer tRNA molecules. Perhaps you're remembering the fact that in bacteria they might have 30 to 40 different tRNAs, and so that explains why they can get by with fewer tRNA molecules and yet still specify 61 codons and convert that into an amino acid sequence. You may be remembering that in mammalian systems we have up to 150 tRNA molecules. So that's a great deal more than 61. So what is the purpose of having those extra tRNA molecules? It's not entirely understood at this time, but some of those tRNAs may specify some amino acids other than the common 20 that we're familiar with. In our next video lesson, we want to look at the general structure of the ribosome. We'll see it's composed of both RNA and protein, and we want to see the relative proportions of those two components. And we'll see how many tRNA binding sites are present in the ribosome.